Start. So today we'll start our new module, microbiology, basically the study of microorganisms. Here, first of all, we'll discuss how the different organisms have been classified. So Carl Weiss in 1977 has given a basic classifications where he divided all organisms into three different domains. The first one being archaea, also known as archaebacteria. Second is eubacteria, which are also known as bacteria. And third is known as eukaryota, also known as eukarya. So these are the basic domains into which all the organisms were being classified. So what are these? We'll discuss. So basically, based on whether there is a nucleus and the other organelles are present or not, we are trying to classify the organisms. So if you look into the basic classification kingdoms, mostly all the organisms have been classified into two categories. One is prokaryotes and other is eukaryotes. The prokaryotes, actually, the domains archaea and eubacteria are actually commonly grouped into prokaryotes because they are lacking nucleus and the other membrane bound organelles. Well, in eukaryotes, they have the nucleus and other membrane bound organelles, and these domains are actually then classified into different kingdoms. Okay, so kingdoms in eukaryotes consist of fungi, protista, plantae, and animalia. Because of the different characteristics, we are classifying them into different kingdoms. So we'll discuss what are the features of this kingdoms specifically. Basic difference between prokaryote and eukaryote, I guess all of you are aware of because you have learned it in your cell biology class. Basic difference is the presence of nucleus in eukaryotes, which is absent in prokaryotes. Basically, prokaryotes are the very small organisms and eukaryotes are the complex or the large organisms. The prokaryotes basically includes the bacteria, while eukaryotes consist of algae, fungi, protozoa, plant, and animals. The size, as I said, it differs. And basic structures, there are different nuclear structures which are actually present in eukaryotes. For example, the nucleus, which is present in the eukaryotes, but it is absent in the prokaryotes. Similarly, in prokaryotes, we have haploid or the single DNA, while in eukaryotes, with the diploid DNA. And then other organelles are present in eukaryotes, which are actually absent in the prokaryotes. So when we are talking about the domain archaea, archaea actually means ancient bacteria. So this is a picture which has been taken from a book. So this is a aerial view of a hot spring at Yellowstone. You can see here I'm discussing about archaea. So archaea, the features are unicellular. They have just one cell. They are prokaryotic, means they are lacking the nucleus and the membrane bound organelles. They could be autotrophic or heterotrophic. Autotrophic means they can prepare their food. Heterotrophic means they are depending on some other organism for their food. And these organisms actually reproduce asexually. So there is no sexual reproduction. And most of the one common feature of this domain bacteria is that they are adapted to extreme climates. Extreme climates in the sense that if they can survive in very hot environment, just like I was showing in this picture, is a hot spring picture where this bacteria can survive. So, for example, in hot spring and geysers area, this kind of bacteria can survive. And since they can survive in this kind of hot environment, they are also known as thermophiles. And some of them can also survive in extremely salty environment. For example, the Great Salt Lake in Utah in United States. This type of bacteria are known as halophils since they can survive in the extremely salty environments. Now, the domain archaea is just one kingdom known as archaea. Moving to the other one, the domain eubacteria. In eubacteria, U means true bacteria. So they are basically again unicellular in nature. They are also prokaryotic. They can be autotrophic or heterotrophic. This organism also reproduce asexually. And the most abundant organisms on Earth are the eubacteria. because this bacterium can give rise to around 10 millions more bacteria in 24 hours. In just one day, they can do through different reproduction, like asexually, they can just divide and form around 10 millions of bacteria. So they are found almost every habitat on the earth. And this Eubacteria has also one kingdom, which is known as Eubacteria. Now coming to the domain Eukaryota, or known as Eukarya, they are actually the eukaryotic organisms. They are very diverse in nature. They could be unicellular or multicellular in nature, and they could be autotrophic or heterotrophic. They can reproduce both asexually or sexually. So in earlier two cases, it was only asexual reproduction, but here we also have the sexual or asexual reproduction. And this domain is now split into 4 kingdoms known as Protista, Fungi, Plantae, and Animalia. We'll be discussing about each of these different kingdoms. So basically, Protista, when we talk about, they are the unicellular and microscopic organisms, could be heterotrophic or autotrophic, and can be infectious in nature. For examples, the amoeba, unicellular algae, plasmodium, which actually causes malaria. The picture is actually has omitted this letter sentence. Slime molds, water molds. These are some of the organisms that comes under the Protista kingdom. Moving to the second one, the fungi. Fungi are actually multicellular organisms. They are all heterotrophic, means they cannot make their own food. They are actually depending on some other organisms. Mostly fungi are actually decomposers, and they could be some infectious agents as well. The examples include the mushrooms, athlete's food, bread mold. Moving to the another kingdom, Plantae. These are actually multicellular green plants that we are looking around in our gardens and all. So these are autotrophic, means they can prepare their own food, and the process is known as photosynthesis. They also have a cell wall. For example, mosses, ferns, trees, flowering plants, they come under the Plantae kingdom. The fourth kingdom is Animalia, which actually has multicellular or animals. And as you know, animals are heterotrophic. They cannot prepare their own food, and also do not have a cell wall. So basic difference between Plantae and Animalia, all know, because one is autotrophic, one is heterotrophic, one has cell wall, other doesn't have any cell wall. Examples in Animalia kingdom includes insects, spiders, crabs, birds, and also we humans are also classified into Animalia kingdom. So summarizing this thing, so we have basically 3 domains: Archaea, Eubacteria, and Eukaryota. Under Archaea, we have Archaea kingdom. Under Eubacteria, we have Eubacteria kingdom, just one kingdom. And under Eukaryota, we have 4 kingdoms: Protista, Fungi, Plantae, Animalia. So this was the earlier classification of the different organisms. Now in 1967, Robert Whittaker actually introduced this 5 kingdom classifications. And in 1988, Margulis and Squash modified the 5 kingdom classification of the Whittaker. They also considered the genetics along with the cellular organization and the mode of nutrition in the classification. So what are these 5 kingdoms? The 5 kingdoms include, first is the Monera. So instead of those things, Archaea, Eukarya, and Protista, and all those, they have here 5 kingdoms, which are known as the Monera, the basic ones, then the Protista, and then we have Plantae, Fungi, and Animalia. So we'll discussing all these 5 together. So kingdom Monera actually includes the prokaryotic organism, the primitive one. That is including the Archaebacteria, which can survive in extreme environmental condition, and also the Eubacteria, which is also known as the true bacteria. So basically, the first 2 categories, Eubacteria and Archaebacteria, are included into the kingdom Monera. So characteristics of Monera kingdom is they are prokaryote in nature, unicellular, autotrophic or heterotrophic, and asexual reproduction takes place. Kingdom Monera. Archaebacteria, as I said, they can live in very hot acid habitats like 60 to 80 degrees Celsius, and also in pH 2 to 4 like the hot springs. Eubacteria, although the true bacteria or the modern bacteria, they inhabit almost every known habitat. And right now in my laptop, in my hands, everything, they are bacteria are present. So these are the consumers, they are actual producers and decomposers. Some may cause diseases, but most of them are harmless. So these are the characteristic feature of Monera kingdom. Moving to kingdom Protista, this kingdom consists of the eukaryotes, mostly unicellular and generally larger than the prokaryotes. Could be heterotrophic or autotrophic, and mode of reproduction is asexual. There are mainly three types of Protista: algae, protozoans, and okay, so the other one is missing over here. So there are three types of Protista here written. I don't know. There is algae, protozoans. Moving to the other kingdom, the fungi, which has the characteristics like they have the eukaryote cells. They could be unicellular or multicellular, and they have the they are heterotrophic in nature, means they are dependent on others, and mostly they go for the asexual reproduction. They live on the organic materials, secrete digestive enzymes, and absorb organic molecules that are found by the digestion by enzymes. So most of the fungi are decomposers. Example includes mushrooms and the molds. You must have seen when you keep a slice of bread out for a couple of days, you see there is like black kind of thing that happens over the bread. So there are no those of molds. Their molds have grown. And mushroom, I believe all of you have eaten. I mean, those who do not have any allergy. So coming to the kingdom Plantae. So Plantae characteristic features are they are eukaryotic in nature, they are multicellular, they are autotrophic. As I said, they can prepare their own food. Both sexual and asexual mode of reproduction is there. Example includes mosses, ferns, and flowering plants. And the fifth kingdom, that is Animalia, that has the characteristic features that they have eukaryotic in nature, multicellular, heterotrophic food habit, internal digestion takes place, and mode of reproduction is sexual in nature, the animal kingdom. So this says something I have actually kept for you, so you can have a comparison of all these five kingdom in one glance. So Monera, you know, cell type is prokaryotic. Other four cases, Protista, fungi, Plantae, Animalia are eukaryotic. Nuclear envelope is absent in Monera only. Rest all the four kingdoms it is present. Cell wall, the different things are there. Like in Monera it is non-cellulose, while it in Protista in some cases it is present. Fungi, they have a chitin cell wall, and in Plantae it is a cellulose, which is which is a polysaccharide, and in Animalia it is absent. Mode of nutrition, autotroph or heterotroph in Monera. Similar here, Protista also auto or heterotroph. Fungi mostly they are heterotroph. Plantae they are autotroph using the photosynthesis mechanism, and animals are heterotroph. Now multicellularity, if they are multicellular or not. So mostly Protista, Monera is unicellular, they are not multicellular, and in Protista it could be anything, but mostly unicellular, and rest three, fungi, Animalia, and Plantae are multicellular. So in today's chapter, we will discussing mostly about the bacteria, which are actually unicellular or microorganisms. We are discussing about the microorganisms, and bacteria are the unicellular. The size is very less and very small, and but they are having a variety of shapes, and the growth patterns and metabolic characteristics allow their different classification of this bacteria. So if you can look, this the first picture is a human hair that has been zoomed over here, and you can, when you zoom it, you can see that you can compare this hair and this small things that are, this green one is the pollen grain, while this purple one is the lymphocyte, and this brownish one is the yeast cell, which again when we zoom it we'll find that the bacteria are in very small size. So this green one is the E. coli bacteria, this yellow is the staphylococcus bacteria, and then we have small viruses, the Ebola virus and the rhinoviruses, which are very small, 30 nanometer. So The gross morphology, if you look how the bacteria look like, how the shapes look like, so they can be of different forms, different shapes. Something there's some maybe of rod shaped, on coccus shaped, or spirals. So, and they also can form in characteristic groups, so aggregates, like they can be in pairs or they can be chains, they could be in clusters and all. So basically basic three shapes are there. Coccus means they are spherical in nature. Bacillus refers to the rod shaped ones. So when we have said coccobacillus means something spherical and with rod shape. So they are very short and plump kind of thing. Vibrio means gently curved. And then the third category is the spiralum, means they are like something like a comma shaped structure. They are like helical, or comma could be a twisted rod. So then again we have spirochete, which is coming under spirillum, and which is something that looks like a spring. Like I'm showing the pictures. Here, for example, Bacillus anthracis, something rod-shaped bacteria. Staphylococcus aureus is something coccus, the spherical ones. Pseudomonas aeruginosa, it is again a coccobacillus. Physiopum bacilli, this is also a coccobacillus kind of thing. Spirillum volutans, like they are spiral in nature. Borrelia burgdorferi is again a spirillum. And Vibrio cholerae is again a spirochete, like Vibrio types shape in nature. And as I said, this bacteria, the coccus, then the bacillus, vibrio, spirillum, spirochete, and then there are branching filaments. These are the shapes of the different bacteria. Now, when we are discussing about spiral shaped bacteria, there are two categories, Spirilla and Spirochetes. So, since Spirilla, they have a rigid helix-like structure, but in Spirochetes, it is a flexible helix. So, it is curved, but it is kind of flexible, and there are different modes of locomotion of different bacteria. You can learn more about it from this thing. So the arrangements of bacterial and how they actually divide after division, how they are actually attached to each other. So when they are cocci shaped bacteria, so they can be on single one. They could be on like in pairs, which are known as diplococci. And if they are in groups of 4, they are known as tetrads. They could be in irregular clusters. They could be in chains. They could be in cubical packets, which are also known as sarcina. And then bacilli again can be of 2, like they could be diplobacilli, or could be in chain like structure, or it could be palisades. For example, here you can see after this bacteria has been divided into 2 diplococcus, and then they have actually formed in a chain like pattern. So it is like a many cocci in chains, for example. And here the division takes place in perpendicular way. So tetras are being formed, and then again the bacteria divides and it forms like cubical like of structure, this packet, it known as sarcina. And if the division takes place in several planes, so some irregular clusters are being formed, which is seen in staphylococcus and micrococcus. So again the arrangement, coccobacillus, bacilli, diplobacilli, as I said the name, diplo means 2, palisades like in the line ways, and streptobacilli in again chain like form. So moving to a typical bacterial cell structure, I think you have learned about this in your cell biology. Still, I'm again repeating. So this is how a cross section of a bacterial cell looks like. So basically, we have this long structures which are known as the flagella, which helps the bacteria to move for locomotion. Then we have small thin fragments like you know the fimbriae. Then we have a pilus, conjugating pilus, which actually helps in the reproduction. And then we have ribosomes. In the cytocells we have different things, and then outer membrane and the cell wall, which is mainly composed of the peptidoglycan, and followed by the plasma membrane. Basic bacterial cell structure. First we have a cell wall, which is a rigid one composed of many peptidoglycan layers. It consists of an outer membrane, a periplasmic space, and then a cytoplasmic membrane, which actually lacks the spherules. And then we have a cytoplasm in which the 70S ribosomes move. So if you remember, in eukaryotes we have 80S ribosomes. So in prokaryotes we have the 70S ribosomes. Then there are certain organelle known as mesosomes. Mesosomes are the areas in which the cell membrane of this bacteria cell, they fold inward. So the role of mesosome is that they play a role in the cellular respiration. Okay, so in this process, it breaks down the food to release the energy. So mesosomes play a role in cellular respiration. Then we have some other storage granules, which consist of lipids, glycogen, polysaccharides, sulfur, phosphate, and other storage compounds. Now, mainly the major component of bacterial cell wall is peptidoglycan. And peptidoglycan is a macromolecule that consists of sugars and amino acid. So sugars, you means carbohydrates, and amino acids refers to the proteins. So as the name suggests, peptidoglycan. So peptido means you can come there, say that the amino acids are referred to this, and glycan means the sugar part. And these all are assembled structurally like a woven material. So the amino sugar component consists of two molecules. So it could be alternating molecules of NAG and NAM. Remember, I have highlighted in red color. N-acetyl glucosamine, also known as NAG, and N-acetyl muramuric acid, which is also known as NAM. So these are actually alternated present in this amino sugar component of this peptidoglycan. And these molecules are actually cross-linked together by short peptides, which actually help the help to give a strength and structure to this peptidoglycan. And thus the peptidoglycan provides protection to the bacteria and defines their shape. This is the peptidoglycan, how it looks like. I mean, you can see the structure has been shown in small hexagonal way. So N and NAG and NAM are represented in G and M, pink color and green color, linked together alternate way, and this peptide cross links are there. You can see over here again, the NAG and NAM, then intermediate. So here comes a question: how do we differentiate the different bacteria? So like different kinds of bacteria are there, how to differentiate those bacteria? So for that, Danish scientist and physician Christian Gram gave a technique which is known as the Gram stain, which is one of the useful and most widely employed differential stain in bacteriology. So it actually divides the bacteria into two groups. One is gram negative, one is gram positive, based on the name of the scientist. So, and the classification criteria is the cell wall, because how actually the difference in the cell wall actually gives, due to the difference in the cell wall, the staining processes gives different results. For example, you can see over here this thing. So gram stain, first of all, we do not know which bacteria we have, and I am using the gram stain. First, I will fix my slide which contains the bacteria. I'll use the stain known as crystal violet. Crystal violet will actually color these cells, so it will look like in this color. Post that will be giving iodine treatment, and iodine treatment will actually give decoloration. A counter stain when they are given, when we are getting the purple color, then we can say that it's a gram positive bacteria, and if it is turning into a pinkish or reddish color, then we can say it's a gram negative bacteria. So we'll discuss this technique in detailed way. So this is how a gram staining actually has happening. First of all, we are using this dye known as crystal violet. First step, the primary stain is the crystal violet, which is actually when given, it will give all the bacterial, all the bacteria into purple color. This is a primary stain. The crystal violet is the basic dye, and when we are putting this dye, we are staining with this dye, then we are getting purple color for all the bacteria. The second one is, we'll be following this treatment with the iodine solution. So iodine solution, when we are giving, it works like a mordant. It will actually increase the interaction between the bacterial cell and the dye, so that the dye is more tightly bound and the cell is more strongly stained. So once we give the iodine, all the cells again it will retain that color and will be looking at like purple in nature, purple in color. Now the third steps involve clearing the smear or cleaning the slides with the washing agents such as alcohol. Usually it is 95% ethanol or isopropanol acetone. So the gram-positive bacteria here will actually retain the violet iodine complex when washed with the decolorizer. Whereas the gram-negative will actually lose their crystal violet complex and becomes colorless. Okay, so here after putting alcohol, you can check what is the color changes happening. So some bacteria will retain the color while some will lose. So those who are retaining are known as the gram positive, and those who are actually removing those color, washing that color is gone, then those are known as the gram negative bacteria. So this is followed by another step which is known as a counter stained method. So we are actually smearing the, we are putting a counter stain with another basic dye which is actually known as safranin. So this counter stain, what will it do is that it will color those colorless bacteria which has actually removed this purple color. They are colorless now. So to look how they are looking, so we are putting this counter stain and it will be so dark. It will be like something of kind of pinkish or reddish in nature. It will give that color. So these are known as the gram negative. And this, after putting the saffron in, it won't alter the purple color of the gram positive. So gram positive will remain the purple. So all through this process, the purple color remains okay for the gram positive, while the gram negative loses this color. It cannot retain the purple color. Rather, at the end, when we are putting some counter stain, the gram negative is becoming red or pinkish in nature. So end result is that gram positive are deep purple in color, while the gram negative are reddish or pinkish in color. This is example how the bacteria looks under a microscope. The purple one, the left hand side is the gram positive bacteria, and the right hand side we have the gram negative bacteria which looks in red color. So why does actually that happens? The science behind this, which actually the gram staining is actually taking place. Why some cells are retaining the color and some cells are losing the color? It actually depends on the composition of the cell wall. So in gram positive, the cell wall actually consists of several layers of peptidoglycan. So you can see in this picture, there are many layers of the peptidoglycan molecule. So this thick layers of peptidoglycan actually helps to support the cell membrane and also provide a place of attachment for other molecules. And these layers actually help this gram positive bacteria to retain the crystal violet dye during the staining method. Okay, and at the final end, we are seeing them, they are purple in nature. So basically the dye is actually trapped in this thick layers of peptidoglycan, and that's why they are actually giving that purple look at the end of the staining method. Well, in case of gram negative, so peptidoglycan is actually a very single and thin layer. So you can see this is the peptidoglycan molecule. We can see very thin layer, and this thin layer actually could not retain the initial crystal violet color, but it actually picks up the pink color of the counter stain during the gram staining. So this method actually, that's why we are getting the reddish or pinkish color in the gram negative after the staining method. So one more picture from the gram positive and gram negative bacteria. The cross section of the both the bacteria, you can see how this thick layer of peptidoglycan is present in gram positive, which is actually retaining the dye, while it's a very thin one in case of gram negative. And also some acids, teichoic acid and lipoteichoic acids are found only in gram positive bacteria, and while these are absent in gram negative one. So sorry, yeah, and lipopolysaccharides are actually found only in gram-negative bacteria, which is another feature of this gram-negative bacteria. Some of the examples of gram-negative bacteria are the E. coli, Klebsiella, Salmonella, and Pseudomonas. While in case of gram-positive, we have Staphylococcus, Streptococci, Bacillus. Okay, so I have also listed the differences between the gram-positive and gram-negative for you. You can see here, over here, the cell wall thickness, and then the peptidoglycan layer, which is actually multi-layered, thick one in gram-positive, which is thin in gram-negative. Now moving to the different structures, other structure organelles of bacterial cell. So as I said initially, they have certain long wires-like structure. So these are known as the flagella, which are actually the organs of motility, and they consist of a protein known as flagellins. These are polymer proteins. So actually the flagellum moves by wheeling about its long axis, helps in movement of the cell, of the bacterial cell. And the number and arrangement of the flagella on the cells are actually diagnostically useful, in the sense that you can, when you are going for some diagnostic tools and all that, so based on the how many or how the arrangement of the flagella is, you can just identify that bacteria. Then there are some other structure known as fimbriae or pili. So these are some filamentous slender hair-like structures, which are again composed of proteins that extend from the surface of a cell and could have many functions. Pili are actually longer than the fimbriae, and they actually help in adhesion to the host surfaces. The third one is the capsules. So most of the bacteria actually they form a thicker and outer capsule of which could be high molecular weight and something like viscous polysaccharide gel like things, and could be some slime layers. So basically this capsule is considered a virulence factor because it actually helps or enhances the ability of the bacteria to cause any disease. So capsules, if there it is present in a bacteria, so it will be conferring the resistance to phagocytosis. So the bacteria could not be eaten up by the different other cells. So it helps the bacteria to enter and not enter, but it will help the bacteria to cause diseases because it could not be eaten up by other cells to phagocytosis. Now then we have some cytoplasmic structures, for example, plasma membrane. The plasma membrane in bacteria is actually composed of protein and phospholipid. The ratio is 3:1. So it helps in different functions like transport, biosynthesis, and energy transaction. Organelles includes ribosomes. We know 70 as ribosomes are present, which are actually the metabolic reserves. Then we have, in certain cases of bacteria, we have something known as endospores. So Bacillus and Clostridium species in bacteria, they can actually produce some endospores. So these are some bacterial survival structure that are actually highly resistant to many different types of chemical and environmental stresses. So these are some resistance molecules. So they are usually formed intracellularly and contain a genome and all essential metabolic machinery. So what exactly happens is that the endospores is actually encased in a complex protective spore coat. So in different other habitats like very high stress condition or high heat conditions, though endospores actually helps the bacteria to survive those kind of environmental situation. So endospores actually are inert resting cells produced by some gram-positive genera, example like Clostridium bacillus and Sporosarcina. So they have actually two phase life cycle. One is the vegetative cell and one is endospore. So vegetative cell is actually metabolically active and growing, while the endospore actually, when exposed to advanced environmental conditions, they have this capacity of high resistance and actually long-term survival. So sporulation means these endospores are formed, and they are the hardiest of all life forms. They help the bacteria to withstand extreme heat, climate, drying, freezing, radiation, as well as the chemicals. But these do not mean that they can reproduce. Now in germination mode, what happens? This bacteria again return back to their vegetative growth. So spore forming bacteria are very resistant to lysozyme, heat, radiation, drying, and can remain dormant for hundreds of years in nature. So and once these conditions again return back to the favorable time, that's favorable for growth, the spores then germinate and return to the vegetative state. So aerobic bacillus group and anaerobic clostridium actually develop some endospores. They are widely distributed in nature. They are also present in the intestine of humans and animals. So this endospores actually helps this bacteria to survive different environmental adverse conditions of the environment. And moving to the bacterial, some of the internal structures. So as I said initially, there are some inclusions or granules. So there are some intracellular storage bodies which actually vary in number, size, and also content. So bacterial usually use them for environmental sources. When the environmental sources are depleted, so they use that from that storage body, whatever the energy. For example, glycogen, polyhydroxybutyrate, gas vesicles for floating and all, etc. So these things, the bacteria can use this storage bodies when there are conditions are not good for the bacteria. And if I summarize these things, so we have a cell wall which is actually protecting the cell or bacterial cell and also gives a shape, the cell wall. Followed by a cell membrane or plasma membrane, which is actually regulating the movement of materials into and out of the cell. They also contains the enzymes for respiration. The cytoplasm, actually the main thing where we have the DNA or the ribosome and the other organic compounds. So, and we have the chromosomes which are actually carrying the genetic informations which is being passed from different generation to generation. Then we have something known as plasmid, which actually contains some genes that are obtained through the genetic recombination. And we have a capsule, which is or a slime layer, which actually protects the cell from different immune attack and also helps in assisting the attaching the cell to other surfaces. And as I said, no, endospores, they are present in some kind of bacteria to protect them from different harsh environmental conditions, could be heat or drought. Then we have pilus or pili, so they help to attach to other surfaces, also help in genetic recombination. And finally we have this big longer thing one, that is the flagellum, that helps in locomotion or movement of the bacterial cell. So up to this part, that was to I had to teach you. The rest part ma'am will be teaching you, covering the other microbiology part. So CRs are requested to communicate with Monipa ma'am, and she will actually finish the rest part of microbiology. And the sixth module, as I said, will be taught by Professor Surabhi Chaudhary. So yeah, someone has replied. Divyanshu, what he has said, but nothing. So yeah, so yes, so next Tuesday class onwards, I will talk to ma'am, and mainly she will be starting the sixth module, and Monipa ma'am will continue with her